Hey A&Pers, this is Dr. Jones checking in. This is A&P 1, Lecture Test 1, Video 13, Lucky 13. And we're on page 21. Video 13, NoteSet page 21, which for me looks like this, centrosomes and centrioles. All right, let's dig in. So I mentioned in the previous video, we talked about this in great detail. Okay, we got the basic concept in great detail. We will add lots more detail to it throughout the semester. So this is information, that's a star, does not go away. I told you I wasn't an artist. All right. So the cytoskeleton, cyto means cell and skeleton means skeleton. So supports the cell, holds it up, holds it inflated as it were. Okay, and then we talked about the microvillus, microvilli, and we introduced our first Latin endings. So I will not test you over the Latin endings. I will give you Latin endings as we go through and a few Greek endings as we get there. Not because I'm going to test you over them, but because if you know what these are, then our book is really bad about introducing like the singular and then 3 pages later using the plural like you know what they mean. Or vice versa, using the plural for a page or two and then 3 pages later they use the singular. And if you don't know those Latin endings, then you're like looking up in the index in the back of the book. Hey, there's an index in the back of the book with like terms and page numbers and stuff. Handy. And it's handy to look up the first occurrence in the book because that's where the definition is likely to be. There's also a glossary in the back of the book with some definitions of some of the terms, not every term in the book, but a lot. And so I give you the ending, singular, and plural just so that you don't get as confused when you see a new term. Although I will say confusion is part of the learning process. So if you're not ever confused, you might not be doing it right. Like the confused and frustrated, that's like the step right before the oh. And if you don't like push through that, you won't get to the fun part where you like understand things and they make sense. So I know we're kind of not raised really to like live in confused frustration, but it is an important part of the learning process. So don't let the confused and frustrated stop you, because it's normal. And this is a hard class, and that's just part of it. So, I mean, if you do manage to get through this class without ever being confused or frustrated, I'm not going to say that you're wrong. That's just astounding. I don't, if it's ever happened before, I didn't hear about it. Let's put it that way. Not that it's impossible. Like I had friends who could like never go to class and like just read the book once and then get an A on the test. Like, and I'm bright, but I'm like book smart bright, and I still have to study my butt off. So most of us have to study and be frustrated and confused. Here's a study hint that worked for me. When I would read the book, I would read the chapter. As I did this, I remember doing this especially in biochem because biochem is not my thing. And so I was shoveling information into my head and so I would read the chapter, but not just eyes skimming over the words, okay, I read it, but spending the time to try and process what in the high holy Hades are they talking about? The confused and frustrated bit. And really spending time just trying to wrap my head around what does this mean? And anything that I could not understand the first time I read through the chapter, And your chapters are bigger, so I would go in sections. I would highlight in yellow. So then the second time I read the chapter, I would review the things that weren't highlighted because I understood those parts and I just needed to start reviewing them so that I'd remember them by the time we got to the test. And I would focus on those yellow parts because the yellow parts were the things that I had not wrapped my head around yet. And I would spend time reading, rereading, looking up in the index, in the glossary, Googling things. Okay, Google wasn't a thing at the time, but trying to make it more relevant for y'all. Y'all can Google things and YouTube videos and all kinds of crazy stuff that we did not have when I was in school. Fun, fun. Not bitter, not at all. But the parts that still didn't make sense, I would highlight orange. So then the third time I read the chapter, biochem is not easy. The third time I read the chapter, I would review the stuff that wasn't highlighted and the stuff that I had highlighted yellow, because those were things I had at this point wrapped my head around. But I would really dig into that orange section and do some more reviews and look up some more sources and ask my friends and ask the teacher and like just reread it, like Veronica Mars, like how many times you have to read it till it makes sense. Just kept focusing on that part until I could make some sense of it. And the stuff that I still literally could not wrap my head around, I would highlight in green. And then the fourth time I read the chapter, I would, before the test, I would review the stuff that wasn't highlighted and was yellow and orange. And then the green parts, I would memorize the words in the order they went in. Biochem, it's kind of necessary. Okay, so it's not easy. It does take time and energy and determination. But you can do it. And even if, here's my thing. My thing is, as long as you are learning at capacity, I am happy at you. When my daughter was little, because you'd be like, I'm mad at you. She'd be like, I'm happy at you. And I totally think that should be a thing. Like, that should be something we say to each other constantly. So it's something I sometimes say. I blame my daughter. But if you're learning at capacity, I'm happy at you. If you are doing your absolute best, then we are square. Don't ever apologize to me for a grade. I firmly believe what's at the 4 tenets, and anyway, the fourth one is always do your best. And there may be days where your best is different than other days. Like what if you have the flu or cold, right? Your best is not the same as when you're healthy and happy and hale, which means healthy. And so always do your best and always just keep doing your best and accept that your best that day is your best that day, and it might be different than your best on a different day. And that's okay. As long as you are doing your best. So for me, as long as you are learning at capacity, as many new words as you can learn in a day, as long as you are putting in the time and energy for you to learn that many new words every day, then I'm ecstatic that you're learning things. So lots of people take this class a second time. It's not easy for everyone. For some people it is, and that's fun. But it's worth doing. So I'm proud of you. So keep on keeping on. All right. All right, we're trying to talk about organelles. All right, so we're at the top of page 21. I will get back on topic. Centrosomes and centrioles. So the centrioles are what I call the Christmas candies, although nowadays I think the Twizzler nubs is more the appropriate reference. I don't think Christmas candies are a thing anymore. Let me get my face out of the way. There we go. Okay, so centrioles are 2 cylinders and they lie perpendicular to each other. So here's the math symbol for perpendicular. It means at right angles. This is the symbol for parallel, which I think is fun because A, they're parallel lines, and B, parallel has two L's together in the middle. So, and during cell division, fill in the blank, cell division, the centrioles form the spindle apparatus. And here's the thing, this one has three different terms that all mean the same thing. They form the spindle fibers, which are these individual fibers that they make. They're also called all the fibers together are called the spindle apparatus, which is this, all of them together are the spindle apparatus. And they are formed during mitosis, which is cell division. So they are also called the mitotic spindle. So spindle fiber, spindle apparatus, mitotic spindle are all correct answers. And if you see any of those three on the lab test, you do need to recognize that those are all three the same thing. Okay. Centrioles, cells without centrioles cannot divide. Some examples: red blood cells, skeletal muscle cells, cardiac muscle cells, and your typical neurons. So without centrioles, because they're important for cell division, without them, you cannot divide. Okay. And centrioles are found in an area of the cytoplasm called the centrosome. All right, so let's talk about flagella and cilia. And cilia are, okay, here's a cilium. And these are microvilli. A cilium would be much longer. The cilia are longer than microvilli. Okay. The cilia are long, slender extensions of the plasma membrane, as opposed to the microvilli, which are short, slender extensions, like fingers. Motile cilia, which are the ones that beat. There we go, beat. Like that. Motile cilia are located lining the respiratory and reproductive tracts. The respiratory tract is the breathing, and the function of the cilia there are to sweep up the crud, the mucus in your lungs. And the motile cilia function to move stuff across the cell surface. They sweep up the crud from the lungs. So what happens when you smoke, either way, smoking is bad for your lungs. Partly because it like paralyzes the cilia and it stops that mucociliary escalator. They just changed the name. Used to be the ciliary escalator, but they changed it. And so you're no longer sweeping up the crud out of your lungs. Now it's just sitting there. So then you stub out your cigarette and you say, well, I value my health, or I want to be there for my kids, or smoking smells bad, so I'm going to quit. And you stub out your cigarette and you never light another one. However it works, everybody processes these things differently. However it works for you. And then those little motile cilia wake up and they start sweeping stuff across the cell surface. And they start sweeping up this nasty, gross, disgusting crud that you're now coughing up and spitting out. And then you think, you know what? I was healthier when I was smoking. And then you light up again. No. I'm going with Hagrid on this one. Better out than in. So it's better to have functioning motile cilia, getting all that crud up out of your lungs, instead of letting it fester in your lungs to possibly get infected, or at the very least block some of your gas exchange surface, because that's how you get oxygen to your body, and the oxygen is how you make ATP, and that's kind of what keeps you alive. Third blank, so you know I'm serious. So the motile cilia. Oh, a singular for cilia. is a cilium. 1 cilium, 2 cilia. Same with flagella. 1 flagellum, 2 flagella. So the flagella are whip-like extensions of the plasma membrane. The flagella are like this long, skinny. I can't draw. You know I can't draw. All right, so they're long, thin, whip-like extensions of the plasma membrane, and the only human cell with a flagellum is the sperm. So that's what they look like. That's a flagellum. Okay. 1 flagellum, 2 flagella. 1 cilium, 2 cilia. Now what was the last one we did? 1 microvillus, 2 microvilli. All right, let's finish this page. Ribosomes make proteins. They are organelles responsible for protein synthesis, which is jargon for they make proteins. There are small and large ribosomal subunits. They snap together around the mRNA to make a protein. And I don't remotely care about the large and small ribosomal subunits, but they're in the picture, and so if we don't address it, it is confusing in a way that we don't need, because they're not that important. So basically, there are 2 parts of the ribosome that work together, and you have to have one of each to snap around the mRNA, or it won't work. Okay. So the ribosomal RNA, rRNA, is the majority of the ribosome. 60% of the ribosome is rRNA, and the other 40% of the ribosome is protein. The free ribosomes are in the cytosol. They're floating around in the cytosol, and they make proteins for use inside the cell, which I'm pretty sure is down here in like another 2 sentences. And then fixed ribosomes are attached. See, here's some free ribosomes. There, it draws the second time. And then these are fixed ribosomes because they are affixed, which means attached, to this rough endoplasmic reticulum. And they will actually make the protein into this little chamber, little cistern here. And so those proteins made by the fixed ribosomes are for use outside the cell. So ribosomes are made of, yeah, we already said this, 60% rRNA and 40% protein. And ribosomes make proteins. I already said that. Which ribosome makes proteins for use inside the cell? There was a movie called Free Willy, and it was really terrible. And the sequels were worse. And I think of it every time we get to free ribosomes. And which ribosomes make proteins for use outside the cell? And those are the fixed ribosomes. And explain why liver cells need more ribosomes than fat cells, which are adipocytes. So liver cells make lots of enzymes. Lots of enzymes. What are enzymes? We said this in chapter 2. The enzymes are all proteins. Not all proteins are enzymes, but all enzymes are proteins. One type of protein, enzymes. Okay. So liver cells need lots of ribosomes to make lots of proteins, but adipocytes are mostly for fat storage. And so they don't make as much protein. They don't make much protein. So that's why. So let's finish page 21, and this is the last of the non-membranous organelles. So proteasomes digest proteins. The protea here means protein, and some is soma, which means body, and they digest proteins. Okay, and proteolytic means protein digesting. Remember, lytic is lysis, which is a loosening of, which can be used to mean a loosening of, or a digestion, or an explosion, which is always fun. We'll get to that. So proteolytic is protein digesting. So it digests proteins. So it has enzymes that digest proteins. And are proteasomes membranous or non-membranous? And they are non-membranous. In fact, all of the organelles that we have talked about so far are non-membranous organelles. In fact, why don't you, at the bottom of the page, draw a line and write non-membranous above it, because all of these organelles that we have talked about in the past page and a smidge are all non-membranous organelles. So then the top of page 22, I have a line at the top of the page, and under the line I have written membranous, because starting at the top of page 22, these are all membranous organelles, which we will talk about in our next video. So this has been A&P 1, Lecture Test 1, Video 13. Did we do all of 22? No, we did all of 21. So we covered page 21. All right. And some study skills, right? We covered study skills. Okay. Yeah, we got all that. Okay, we're good. So thank you, as always, for tuning in. The education must go on. Dr. J out.